Arun Prasad Pandurangan from the MRC Laboratory of Molecular, it's been a long conference, right, at least for the staff, Molecular Biology. And he's going to be talking about the SUPERFAMILY 2.0 HMM library and genome assignment server. Thank you, sir.
First of all, I would like to thank the organizers for giving me the chance. And I thank you all for coming here today. So today I will be talking about a resource called SUPERFAMILY. It's a resource used to annotate proteome sequences with domain structural information. So in the overview of the talk, I will be talking about the general pipeline that we have used in the SUPERFAMILY. And then I will touch upon some of the major features currently the resource has. And I will introduce you to a new website that we are currently developing for this SUPERFAMILY, apart from the old ones that is currently existing. And I will discuss about some of the major features currently in the library, in the website. And I will also discuss about briefly about a collaborative project called Genome 3D that we are involved in relation to SUPERFAMILY annotation. And then finally, I will talk about the future progresses, the current progresses and future plans for this resource. So the SUPERFAMILY annotation pipeline mainly is based on a domain classification database scope. The scope database classifies protein domains into multiple hierarchical levels based on a class, fold, superfamily, and family levels. So taking the SUPERFAMILY sequences as a seed sequence, we would try to build hidden Markov models representing those seed sequences in the SUPERFAMILY hidden Markov model library. For the purpose of generating hidden Markov model libraries, we use well-known programs called Jackhammer and SAM. So these tools take a seed sequence and do an iterative search against the known protein sequence database and gather all known remote homologs in the form of multiple sequence alignments. And from that alignment, they build hidden Markov model libraries. So these models represent a probabilistic way to describe a multiple sequence alignment and the variations present in it. So with this model library, the SUPERFAMILY annotation pipeline, very in abstract fashion I'm explaining here, takes an input sequence, protein sequence, and then it will run through the pipeline where it will search the sequence against the models, and it will produce annotations for you in terms of SUPERFAMILY domains. So these annotations actually serves as a basis for all the other works that we are doing in SUPERFAMILY database and the website, which I will explain some of the features in the next slide. So this website currently exists. It has various important features, including, as I mentioned before, the domain assignments and the hierarchy, where the hierarchy is a set of predicted SUPERFAMILY domains in a particular order from N to C terminal of the query sequence. So having got this sort of domain assignments, one can actually start building phylogenetic trees based on domain distributions for a given set of genomes, as shown here. So this is one of the features we have in our website. Apart from that, SUPERFAMILY website also provides user with a download facility where they can download the whole set of hidden Markov model libraries, as well as the pipeline scripts where they can run their own analysis locally. In case if they have a heavy-duty job on analyzing lots of genomes, they can actually use their local computers. And then SUPERFAMILY website also has a set of comparative analysis tools, genomic tools. One of the major features is the identification of over and underrepresented superfamilies in any given set of genomes. So these representations can also be used to construct phylogenetic trees again, and it has other visualization tools to visualize the domain distributions in a given set of genomes. Apart from structural annotations, SUPERFAMILY also has features where it will try to map ontology terms, including gene, function, disease ontologies onto domains that we have got in the library. So now I will introduce you to the new website, which is in the beta version. Still, it's ongoing project. So this is the homepage of the website where user can access it And then it has a lot of entry points where they can derive information from the database. For instance, they can browse through the set of genomes annotated in the database using the genome browser. And they can also make a keyword search. Currently, the search facilities is in infancy. It can search based on sequence IDs and domain, superfamily domain IDs. But we are working on more sophisticated searches, because that would actually help to actually look at the contents of the database, which is very important. Apart from that, user can also submit their own query sequence through the search interface, where it will go through the pipeline and come up with the annotations for the new query sequences. And then this page describes a genome browser page where user can actually browse through all the genomes currently present in superfamily database. And following these links, they can actually access the domain annotations present in those sequences. So since the list is quite a large one, we have just provided a navigation panel on the right where they can jump onto their desired genome based on the taxa. So I would like to just discuss about the current updates we have done on superfamily database recently. So we took up all the UniProt sequences dated April 2018. We ran through the annotation pipeline, and we produced domain annotations for them. We have got about 64% of the UniProt sequences being annotated with at least one domain in the database, which you can access it. And this is the pie chart describes the distribution of genomes present in complete genomes present in the UniProt downloads. So major fraction of the genomes comes from viruses and bacteria. And the database has currently got over 63,000 genomes annotated currently, which you can actually browse. You can submit the sequence IDs of these UniProt. You would get the annotation. And also we try to update NCBI resource also. We just added around dated. The update was done on August 2017. We took about 93 million sequences, and it composed about 102,000 genomes in it, complete genomes. And most of them come from bacteria and viruses again. And we have got annotations about 67%. So now back into the website interface. This is the landing page for a genome view, where, for instance, here you're looking at a Homo sapien genome annotation. So this page gives you all summary about annotations in terms of sequences, number of domains it has annotated, number of unique architectures, number of unique superfamilies, number of domain combinations you have got, and also about the percentage of annotations for this genome. The user can also download the entire FASTA sequence of this genome through this download option, as well as its corresponding domain annotations. And for this genome, if the user wants to find all sets of superfamilies in the genome, they can just follow on this superfamily tab over there next to the genome information, which is shown in the next page. Which is shown in the next page, where this page describes all known superfamily domain annotations for Homo sapien genome. It just lists everything. They can just type on the search box to refine the search to a specific domain superfamily name. And here it shows that the Homo sapien genome contains about 941 ubiquitin-like superfamily domains coming from 853 proteins, and it has got 144 unique architectures, and has got 7 families in it. So user can follow on these links to further see what are the sequences that contain those superfamily domains. And that will lead you to the sequence page, which is the terminal page of this website, where it gives you the entire annotation for a given sequence. So here we are looking at a superfamily annotation for a Homo sapien sequence. And here you see it has got 3 superfamily domains being annotated on it. And it shows all the regions that fall on the query sequence. Apart from superfamily domain annotations, superfamily also suggest a possible family that represents this given superfamily using a subfamily classification procedure. As a result of this procedure, one can actually identify a closest protein chain that represents a given superfamily. For instance, this superfamily, this protein chain most closely represents this superfamily domain. So this has a consequence of building 3D models. So with this information, one can actually build 3D models for these annotations. So in connection with that, I would try to jump into a collaborative project that we are into on building 3D models, and then I'll be back onto this web interface. So in this page, I'm trying to explain the project. Genome 3D project is a UK project. It's a consortium made up of 7 groups in the UK. As you heard from Michael is also FIRE is also part of this consortium. Superfamily is also part of the consortium. So what it does is that it will try to pick up a set of model organisms of significant importance and try to see what sort of structural information is present inside that. Since we have got multiple partners, so we will all come up with our own annotations and a generation of 3D models. So we wanted to come up with a consensus approach where we can represent our known annotations in one interface. That's the theme of the project. So we will all submit our own annotations, our own 3D models, and the Genome 3D will integrate them into one unified view. In this project, we took up 11 model organisms from UniProt, all UniProt sequences. It has got around 152,000 sequences we took, and we ran through the annotation pipeline. We got around 67% being annotated, and based on that annotations, we can actually build 3D models because we have got the templates. So we built around 175,000 odd models in it. And these models and the corresponding annotation would go into the website later this year. So back into the website. The website also has features where you can actually look at the summary of all known genomes available there, all the summary statistics for the user to have a quick look up. This page describes that. So we classify genomes based on the type of model organisms, strains, and special fields like metagenomics, pseudogenes, and others, where others include UniProt, NCBI sequences in it. And then Superfamily also provide a scope view, a scope domain classification database view inside in it, so that user can actually browse through various levels of classification and look at their protein of interest and make sense out of their interpretation. And on sequence search facility, user can submit an input space sequence in the text box, or they can now upload a sequence. They can upload up to 1,000 sequences, and usually the result comes on the fly quite fast. You don't have to wait for it because the searches are quite fast using Hammer package. And you can optionally submit your input so that you get your results recorded later for the later use as well. And talking about future projects and current progresses. So currently, Superfamily annotation pipeline and its hidden Markov model library can cover 88% of the PDB sequences. So clearly, we are missing some of the models in the hidden Markov model library. We need to expand it to make the coverage better. So in order to do that, we are collaborating with the original scope authors, Alexei, in updating and coming up with new superfamily sequences. Also, we wanted to add other domain classification databases, including CATH, SCOPY, and ECOD. And then we take all those superfamily sequences, try to generate HMM models, and in the process, we are trying to expand the model library. And as a result, if this process is done properly, you would achieve 100% coverage with the current trends of data available in the PDB. that has a lot of consequences in terms of genome annotations later on, because that's what we wanted to see in the genome. If we miss coverage, we will miss information. So we are working on this. And finally, I would like to, still the coverage is going up. So finally, I would like to discuss about the future plans for this resource. This resource is well-established resource, I think well into 10 years old, I suppose. And it has been widely used. Sorry, I didn't pick up the statistics. I should have given it. It has lots of hits as well. I don't know, I can only say lots, but no numbers. But I think it scales up with other, I think FIRE also. So what we wanted to do is to integrate the Superfamily website with two of its sister resources, called D2P2 and DCGo. The D2P2 resource is it's a resource that will take up all the known disorder prediction programs, and it will compute a consensus predictor based on multiple predictions, and then it will represent them on the superfamily domains. And then DCGo is a complete package based on ontology mapping onto superfamily domain. It is domain-centric view, so you map the ontology terms onto domains that you know already in the PDB. So you can actually relate your mapping more onto structure. So the idea behind this is to merge structural information from superfamily and disorder information from D2P2 and the ontology terms mapped onto the domains. It will give you a unified view of the sequence that you're looking at, and you can make better interpretation of it. And finally, I would like to thank Professor Julian Goff and his group for the support and encouragement. I'd like to thank all the present and past Superfamily team for all the developments. And also, I would like to thank my funding body, BBSRC, and my research institute, MRC LMB. And I thank you all for your attention. Michael has a question. Yes, Michael.
You managed to annotate a huge number of sequences. I mean, what sort of computational load? I mean, you're still doing hidden model
Yeah.
which is non-trivial.
The annotations we have got already. So the computation load is so weak. We can generate models quite lightning speed because we use modeler. For example, to produce human genome models, so it takes about 6 hours.
For the whole human proteome?
Yeah, because we have got all the annotations in the database. As and when new sequences come, it's been recorded in the database. And we got the templates there.
So if there's a new sequence, you've got a new proteome, a new eukaryotic proteome.
Yeah.
Say 20,000 proteins, genes.
Yeah.
How long would that take to run?
If it will take a few hours, like 3.
Oh, okay, that's very interesting.
Yeah, but we are not actually proposing that we would build a proper model, but it's a more on an annotation basis. But we could do a better job on model building also.
Yeah,
Hi, Mark.
that's what I was wondering about the model building.
Yeah.
You're building all the models yourself in Modeler. Why not?
Yes.
I was wondering, why not collaborate with FIRE or some other
Exactly,
server
I mean,
that's so specialized more in modeling, I
It's
guess?
totally doable. Currently, we were focused on annotations, but since with the collaboration like Genome 3D, it opens up other possibilities of improving the model and interacting with other groups.
But all the groups in Genome 3D making their own models right now?
Yes,
Or
yeah.
is it a centralized model?
The model making process is different, annotation is different. As a result, the repertoire is quite different. But there must be a consensus. And if there is a consensus, then it has to be right there. So that's the idea. Thank you. Hi.
So for the Genomes 3D, is it only for human or you have other organisms?
Currently, we are looking at 11 model organisms. That's based on scientific advisory suggestions on what genomes to select. But we would expand later on to further important genomes. Thank you.